To show you actually how this has evolved over these years, it's my great pleasure to introduce Susan Desmond-Hellmann to you, who is, Susan is the Chancellor of the University of California at San Francisco, and many of you know her in the drug development world for many years. I would just mention that she was named in November of 2009 as one of the world's 7 most powerful innovators by Forbes. That's quite an honor. For anyone, but in Forbes, that's really an honor. She previously served, as many of you know, as the President of Product Development at Genentech, and I would have to say during her tenure there, they became the #1 producer of anti-cancer drug treatments, 2 that you know well, and we know so well in the cancer world, Herceptin and Avastin. So Dr. Hellmann will now give us some comments, and we'll move on from there.
Good morning, everyone. I'm very, very happy to wake up early and join you from San Francisco. As you've heard, one of the great joys of my life was being involved in the development of trastuzumab, or Herceptin. So I wanted to start my comments by remembering back almost 12 years ago now, when we worked with Dr. Woodcock and her colleagues at the Food and Drug Administration to obtain the approval for what was then the first personalized medicine ever for cancer patients. Remember what was important. Only HER2 positive or overexpressors of HER2 were eligible for Herceptin treatment. And I predicted, as many did in 1998, that this was the way we would do business. But a reality check on that. In fact, biomarkers and subsequently imaging, while incredibly promising, have added complexity, slowness, and cost to our clinical trials process. So one of the fantastic things about coming to academia at a time like this is I think this kind of dilemma is exactly where academia, and specifically institutions like UCSF and thought leaders like Laura Esserman, can add so much value. So I want to tell you why I'm excited about I-SPY 2. We have to ask and answer questions in cancer drug development more efficiently. Most people think about innovation as coming out of the lab, but what we're talking about today is innovation in how we do clinical trials. This has enormous importance for patients with cancer and other diseases. If we can ask and answer questions, we can much more quickly bring the right therapies to the right patients at the right time. So what I-SPY 2 means for me is our ability to take this biology, to take these advances, and much more rapidly bring them to patients. Alternatively, at times we shouldn't bring these advances to patients. The answer, in fact, is no, and we should discard some potential new therapies, again, as quickly as possible, so patients aren't exposed when, in fact, the medicine is either not going to help them or is unsafe. So the reason this trial is so important is all of us have a shared sense of urgency to bring our technological advances to patients as efficiently as possible. And I-SPY 2 for me is a breakthrough in technical improvement in our clinical trials process to change the lives of patients. So almost 12 years after our first personalized medicine, what I-SPY 2 means is that we can much more quickly bring such therapies to patients, so we're not sitting here 12 years from now without far more advances available for not just breast cancer patients, but patients with a variety of different disorders. So I want to salute Dr. Esserman, Dr. Berry, and really all of the collaborators involved in this trial. I think this is a great opportunity for all of us to see something that really has never been seen before. So congratulations to everyone, and I'm happy to join you this morning.